Hey, this is Carson from iSensor. iSensor is a mobile health company that turns smartphone into an instrument for clinical check. So today I'm going to talk about the topic, the diagnosis of DOTA 2 in 30 minutes. Is it possible? Since discovery back in 2014, there are many children diagnosed with DOTA 2. However, there might be more left undiagnosed. They usually have their first diagnosis when they have a stroke and went to ER. The doctor might be puzzled and cannot find the reason until they do a genetic testing. However, this kind of testing might be time consuming and costly. If we can have a testing solution which will allow doctors to get the information timely, that will empower them to make timely decisions. Especially with TNF inhibitor, many children with DOTA 2 actually have their life back in control. So if they can get the diagnosis earlier, they can stay healthy and keep their life in normal. In the meantime, for us to developing such kind of testing for rare disease, the challenge including technical challenge and also economic challenge. Because for this kind of clinical testing, usually it might take 3 to 5 years to develop, and it involves resources and talent from different areas like biochemistry, electrical engineering, mechanical engineering, and regulatory. Therefore, it could be a costly process and lengthy to develop. And considering the relatively small patient population, many big companies might feel hesitate to develop this kind of testing. That could be beneficial and life-changing for patients with rare diseases. I met with Dr. Chip Chambers last year. When he see our device, he say, maybe we can change again with your innovation. Here is a little bit background about what we are doing right now. So iSensor is a startup that founded back in 2012. Since then, we have been working on using smartphone or smartphone-based device and to make tiny measurement more accessible for the population. What we do is we turn the front camera into an accurate optical reader, and also we utilize the screen as a light source. Together, they become a lab-grade instrument. Combined with existing optical strip technology, we can quickly develop a new test and turn it into a cost-effective solution. Take hemoglobin A1C, for example. It usually takes 5 to 7 years to develop a new product. However, with this platform, we developed a new A1C test within 9 months. And the use of smartphone-based system also allows seamless transfer of health information and allow communication with healthcare providers. With this innovation, we get the world's first FDA-approved smartphone camera-based blood test. And since then, we have been expanding the platform for different disease areas. So today, we have product to help patients with chronic disease like diabetes and cardiovascular disease. And also we have a hormone test for people who want to get pregnant. And very recently, we have been working on infectious disease, especially for COVID and influenza. The same technology can also be applied to rare disease. And DOTA 2 is our first trial to make this possible for instant testing for rare disease. So right now, there are existing testing like next generation sequencing, PCR, and ELISA. However, to get an instant result, most people rely on test strip-based test. For this development, it requires significant resource and the involvement of biochemistry development and also engineering. However, most strip developers, they are limited in resources and they can only focus on the biochemistry part. And this is where iSensor can come to help. We developed a PiezoTest X Lab and provide them the meters so they don't have to build the block from scratch. They can focus on the development of optical strip and combine with our meters, and together it becomes a solution. And this kind of co-development model is something we launched last year, and we have been working on a few strip vendors, and one of them happened to be working in the rare disease field. Previously, I have developed the test for PKU, and when I mentioned to them that we are interested in developing a test for DADA2, and they are passionate about developing this together. So here is the workflow we propose for a strip-based on-the-spot DADA2 test. So instead of getting a sample and send to a remote laboratory, the healthcare provider can simply prick finger of the patient, collect a sample, and mix it with EHNA. This will inhibit ADA1, so we only get the AD2 full reaction. And once we get the mixture, we apply it to the cassette. And it takes about 15 minutes to react. Afterwards, the meter can get the results and tell the doctor whether how high is the ADA2 concentration. So before we start this project, we look at what is available on the market right now. So most of this kind of test kits are ELISA-based, which use wet chemical. So we leverage what we know about what is working and try to turn it into a dry phase strip, which is available for point of care testing. So we initiate the feasibility study. The main idea is to turn the wet phase test into a dry phase strip. So the initial investigation is to determine if enzymes and indicators where enzyme will interact with ADA, and the indicator will change color based on the concentration of biomarkers. And we first immobilize them onto a softened membrane to generate signal. And here, this is the process of the overreaction. And for the experiment, we experiment with different indicator, different pH value to find the best combination. So during the past few months, we have been experimenting with indicators like MADB, LPS, those are Trinder. And also we work on with monodie like SH83, TMB, KN404, and to see what would work best with the current ADA level. So initial results reveal that the Trinder with MADB, although this is the first one we tried, but it doesn't provide sufficient sensitivity below 250 micro per liter. And our goal is to have a clear linearity across the range of 200. And all of the monodie we test, they have some kind of background. So we further adjust the condition, then try to get the best signal. So among all the test sites, we finally arrived at two best configuration. One is with the full AAP plus ALPS. It provides a wide range, dynamic range from 0 to 200 units per millimeter ADA. And here you can see that the strip change color across the whole range. However, the difference is not less significant. So it provides a test that can provide a wide range result, but the sensitivity is something that we want to further improve. And this work with at ambient temperature, and you can get the result within 15 minutes. To further improve in sensitivity, we experiment with TMP. And with the incubation, we can amplify signal. So after the ADA is added, we incubate the test at 50 degrees Celsius for 15, 30, and 60 minutes. And through the image analysis, we identify that incubation for 15 minutes, it already gives us good enough results. And it's especially highly sensitive at the lower range, which is an important range we will be looking at for to determine whether that's an ADA deficiency. So here on the screen, you can see that the strip has significant color change, especially at the lower concentration level from 0 to 20. But above that, it might gradually flatten out to above 100. So these two tests, one gives us a relatively wide range, but it's a bit less sensitive. But the TMB1 it's very sensitive across 0 to 25, but it will be relatively flat for the concentration above. To summarize, through this platform, although during the pandemic, we were able to work with our strip development vendor together and develop the initial prototype for the test. And it could be possible to develop a 30-minute dry-phase test strip, but still there are technical challenges to overcome. And without incubation, the best formula we get so far is the 4-way AP plus LPS, which can achieve a sensitivity of 25 units per milliliter across the whole dynamic range between 0 to 200. And through the single application with TMB, a limitation of detection of 5 units per milliliter ADA could be achieved, yet the dynamic range is relatively more limited. So as the dynamic range is broad, we might further improve based on a certain range that is most clinical significant. And looking forward, we will further use a recombinant ADA to protein to do the test and try to change different incubation time and temperature. And hopefully we can find a solution that provides the sensitivity and usability we desire. So thank you very much.